Hello, I'm Quinn Abfalterer, and I will be a junior at New Mexico Tech this forthcoming fall, studying biology and mathematics. This summer, I had the opportunity to work in B10 at Llano under the mentorship of Magoon Shakya and Dean Morales, studying the evolution and diversity of bacteriophage tail fibers. Specifically, I'm studying bacteriophages by mining data from NCBI databases as well as environmental metagenomes in the hope of better understanding the relationship between these bacteriophages and maybe gaining insights to better understand and fight antibiotic-resistant infections for a method called phage therapy. To accomplish this, I used various bioinformatic techniques, many of which new to me, and learned a lot. Currently, the world is facing an emerging public health crisis other than COVID-19. The rise of antibiotic-resistant bacteria that are causing many infections that used to be readily treatable to become life-threatening, or much more tenuous or costly to treat. The overuse of antibiotics over the last 7 years has led to this crisis. Currently, more than 2.8 million antibiotic-resistant infections occur in the US each year, and more than 35,000 people die as a result. And these numbers are only set to increase as existing drugs become increasingly obsolete. Some studies predict up to 10 million could die from antibiotic-resistant infection by 2050 if no major developments are made. Unfortunately, it is unlikely that drug development will be able to keep up with the rate at which bacteria and fungi are evolving resistance unless there's a major breakthrough. Of the hundreds of new drugs developed each year, only one or two go on to become antibiotics, and this is just not keeping pace with the evolutionary rate of drug resistance, as seen in the graph, which goes from the 1960s to 2010, which has only grown steeper in the last decade. This crisis has led to the consideration of alternatives to traditional antibiotics so as to avoid this crisis. Bacteria, however, have a natural enemy, bacteriophages, or just phages for short, viruses that infect and kill bacteria. The application of this to bacterial infections in a clinical setting is called phage therapy. While still not widely adopted in much of the world, phage therapy has some notable successes and potential to be a viable solution to antibiotic resistance and is gaining more widespread attention. The key to phage therapy is the bacteriophage, a virus that infects bacteria. Bacteriophages are highly specific and use special tail fiber proteins on their surface to attach to their bacterial host target. Just as a key is specific to a lock, bacteriophages come in numerous shapes and sizes, as seen in the illustration. There are an estimated 10 to the 31 bacteriophages on Earth, making them the most numerous organism on Earth. They outnumber bacteria by a factor of 10 and live anywhere bacteria are found, from soil, rivers, oceans, and deep within the earth, to the frozen lakes of Antarctica and the Canadian North. Bacteriophages kill bacteria by attaching to them with specific tail fibers made of protein, as seen in the picture. Penetrating the bacteria and injecting their DNA into the host's genome, and then either waits for better conditions to replicate the lysogenic lifestyle, or immediately, lytic lifestyle goes on to take over the cell's function and turns it into a virus-making machine, before bursting open the cell and releasing many new bacteriophages to repeat the cycle. Bacteriophages are very good at their job, and it's estimated that in the oceans, 15 to 40% of the resident bacteria are killed by bacteriophages each day. In doing so, bacteriophages may influence the oceans and perhaps the entire world's food supply by limiting the volume of bacteria available for other organisms to eat. Bacteriophages also likely play a role in the human immune system, as well by killing bacteria in our respiratory tract. Bacteriophages are a major reservoir of microbial diversity and driver of evolution, by rearranging genes and forcing bacteria to evolve to survive. To harness the power of bacteriophages, though, one must start with knowing the host range of the bacteriophage, which depends on the bacteriophage's tail fibers. The basis for my research was the previously mentioned lysogenic bacteriophage and how they incorporate themselves into the host's bacterial genome, as seen in the figure, for a period of their lifetime and replication cycle. This includes the tail fiber genes that encode for the tail fibers that allow them to attach to the bacteria in the first step of their successful attack. During whole genome sequencing of bacterial genomes, resident bacteriophages can be readily detected. These bacteriophage tail fibers in bacterial genomes are evidence of a successful bacteriophage attack, and finding a bacteriophage with a very similar tail fiber sequence could provide a candidate to infect and kill the bacteria of interest, as it possesses the correct key to unlock the bacterial defenses. Here is an illustration of the bacterial life cycle. We began by selecting 157 bacteriophage tail fiber proteins, encoding genes in bacteriophage genomes from a database of bacterial proteins. These genes were selected because they were very similar to tail fiber genes in living bacteriophages, as we didn't want genes that were not clearly of viral origin. This preliminary selection was done using R. Originally, the selection was much larger, but subsequently tail fiber proteins that were more than 70% similar were clustered to a representative protein sequence so as to achieve greater diversity of tail fibers. Using CD-HIT, we analyzed the diversity of the selected database using MegaX to make phylogenetic trees that gave insights into the homology between the sequences and global diversity of the dataset. After selecting the 157 sequences, as seen in this local blast search pipeline, we searched for homologous sequences in environmental metagenomes. The metagenomes analyzed were from diverse environments such as soil, rivers, polluted harbors, both open and deep ocean, and lakes. I had a specific interest in lakes due to their being to some extent an ecological bubble, and in interesting places from tropical, temperate, desert, and Arctic locations. To search for metagenomes, they must first be processed into usable format using NCBI Magic Blast, followed by searching for homologs with tblastn that first translate the metagenomic DNA into protein and then matches the sequences. Many of the files used are very large and take hours to download and search on average hardware and internet. The quality of the hits, as they are called, was assessed by their E-value, expect value, that measures the chance of such a hit in the global system, percent identity, which measures direct similarity between the two sequences, and query coverage, that measures the number of high-scoring pairs in the alignment between the two sequences. Results were analyzed using R. So far, I've been able to search a reasonable sized set of metagenomes and have some early results. There have been a handful of significant hits. However, these have mostly been with a small group of the query sequences, mostly Rhizobium, which is a species of soil bacteria. Notably, there have been a number of hits with query sequence such as Enterobacter cloacae complex, Salmonella enterica, and as seen in the figure, E. coli. The arrow in the figure indicates a query E. coli sequence that was similar to an environmental bacteriophage tail fiber found in a metagenomic analysis of the water in Lake Baikal in Russia. This bacteriophage could possibly be novel and have the potential to infect all the E. coli strains near the highlighted one in the tree. Some bacteriophages are known to have a fairly broad host range, and just these environmental bacteriophages might be able to infect the pathogenic strains we know well. Just how wide the host range of the bacteriophage is and how far away in the tree it could infect is unresolved. These latter hits, though, have all been slightly below the significance threshold being used in this experiment so far, and further analysis is needed. There is yet a lot of uncertainty with bacteriophage specificity and phage therapy as a whole. As yet, only a fraction of the chosen environmental genomes have been searched. In the future, more environmental metagenomes will be searched, and will certainly unearth more interesting results and hopefully more significant hits in the near future. And that is my project this summer at Lano. It's been a very interesting and educational experience for me, even if done remotely through video calls and message boards. I would like to thank my great mentors, Magoon Shakya and Dean Morales, for their guidance and patience. Their input was instrumental in my understanding of the project and the bioinformatic methods involved. Thanks as well to B10 of B Division for providing a good virtual work environment. I thoroughly enjoyed my experience. Thank you.